Hello, everyone. Today we're going to discuss a very important topic, which is how the body regulates the cell cycle. You know that the cell cycle is a series of events that are repeated in the cell regularly, and we have discussed this before, and we have seen how the cell passes through several stages or phases. So it passes through the G1 phase, and then through the S phase. This is the G1 phase, and then through the S phase, and then through the G2 phase. The G1 phase and the S phase and the G2 phase form together the interface or the interphase. And following a successful interphase, the cell goes into mitosis. And we've discussed the phases of mitosis before. We've discussed metaphase, we have discussed anaphase, we have discussed telophase. Of course, we are talking here and focusing on mitosis. So these are the different phases of the cell cycle, and they are regularly repeated in a certain interval of time. That interval of time will depend upon the type of the cell. So there are cells that are very rapidly dividing, like the cells of the epithelium of the skin, and there are other cells that are very slow in dividing, and these cells, of course, have a longer life cycle. Why is it important to regulate the cell cycle? Because we don't need to or we don't want to have a cell cycle that is too slow. If the cell cycle is too slow, then it wouldn't fulfill the requirements of the body, because we need more and more cells, for example, for growth, for repair. And if the cells are not dividing at an adequate rate for these requirements, then this would result in impairment of the cellular functions that are necessary for life. On the other side, we don't want the cell cycle to be too rapid, because if the cell cycle is too rapid, then we're going to have masses of cells that are forming very rapidly in the body, and the body cannot deal with these masses of cells, and this is the beginning of the occurrence of tumors. And these tumors might be benign tumors or malignant tumors like cancer. So what are the purposes of regulation of the cell cycle? The overall purpose of regulation of the cell cycle is to make the cell cycle at an adequate rate. We want to regulate the timing of the cell cycle. Meaning that we want to regulate when the cell cycle starts, and therefore regulate the duration of the cell cycle. And if you regulate the duration of the cell cycle, then you are going to regulate the rate of cell division. In a eukaryotic cell, this is in particular in eukaryotic cells, there are built-in stop signals. What are stop signals? These stop signals know when exactly to act, and since they are called stop signals, then their main function is to stop, to halt the cell, or to stop the cell unless otherwise mentioned, unless they are overridden by the opposite signals, which are the go-ahead signals. So every now and then, at particular moments, at particular timings of the cell cycle, we have stop signals. So the essential, the baseline, the default is to stop unless we have an overriding signal that is a go-ahead signal, and that tells the cell to continue. Why are these stop signals important? Because just like in your personal life, you need to stop every now and then and reconsider your life, reconsider your goals, reconsider the methods you adopt to achieve those goals. You cannot keep running and running in life without pausing. So the same applies to the cell. These stop signals ensure three important things. Number one, that cell division occurs only after sufficient growth. You cannot ask a very small cell that hasn't accumulated enough cytoplasm and enough genetic material or duplicated its genetic material to divide into two, especially if it is mitosis, and that you need the two daughter cells to be similar to the original cells. So it's logical that this original cell needs to multiply everything by 2 before dividing by 2. When you multiply by 2 and divide by 2, this is as if you were multiplying by 1. This means that the daughter cells will have exactly the same number of chromosomes and the same amount, almost the same amount of cytoplasm as the mother cell. So first of all, this cell has to enlarge twice like this in order to be able to divide into two equal daughter cells that are similar to the original cell. The second goal of those stop signals is to ensure that DNA replication has occurred faithfully. And by faithful DNA replication, we mean a correct DNA replication, because as you know, if an error occurs during replication, then we will have a mutation, and that it is also complete. So DNA replication should be correct without errors and complete, meaning that all the set of DNA, meaning the 46 chromosomes, have been duplicated, giving rise to 92 DNA molecules that will later on be considered chromosomes. The third condition, and of course this should be actually the first condition, you have to make sure that enough nutrients reach the cell. And by enough nutrients, I mean enough glucose, enough vitamins, enough fatty acids, enough amino acids, because how can you achieve adequate growth and how can you achieve DNA replication without having the raw material for these substances to duplicate and grow? Where do these checkpoints exist? They exist in specific areas of, or actually not areas, but specific timings of the cell cycle. So we have three checkpoints that are labeled here. We have the G1 checkpoint, and we have the G2 checkpoint, and we have the third one, this one, which is the M checkpoint. The G1 checkpoint, where is it present? It is present at the end of G1, the terminal phases or stages of G1. What does it check for? It checks for the presence of enough nutrients, and therefore for an adequate cell size that has been reached. It also checks for the presence of enough growth factors that reach the cell, because these growth factors, as we might have seen before and that we'll be seeing later on, are important for mitosis, and they also check that there is no DNA damage. We will see this checking of no DNA damage twice, once before the S phase and once after the S phase. Here, we are checking for DNA damage before DNA replication. But here we are checking for DNA damage after DNA replication. The G1 checkpoint is very important because it is called the restriction point. Why is it called the restriction point? Because if the G1 checkpoint discovers that the cell size is inadequate, or that there are errors in DNA, or that not all the DNA molecules have been replicated, then the cell is forced to stop dividing and to quit the cell cycle. At the beginning, this is temporary until the issue is worked out. But in case the issue cannot be worked out, then this G note phase, or the resting state, will be permanent, and the cell will be permanently excluded and will undergo apoptosis, or self-suicide, as we've studied in the cell. And then comes the S phase and the G2 phase, where the cell continues to grow. And the G2 checkpoint is another checkpoint that occurs when. So the question is not where, but when. It is where on the diagram, but when in timing, because the cell cycle is nothing but events that take time. So the best question is, when does it occur? It occurs at the end of G2, and it checks for adequate cell size again, but more importantly, that the DNA replication and synthesis that occurred is not erroneous, that there is no DNA damage. And if everything is okay, then we will enter into mitosis, and comes the third checkpoint, which is the M checkpoint here. And the correct place of the M point is between metaphase and metaphase and anaphase. And the M checkpoint checks that the chromosomes are adequately attached to the spindle fibers, because you have here, this is the centromere where the two sister chromatids are attached, but here we have the kinetochores, and these are disc-like proteins where the spindle fibers, which are essentially made of tubulin, are attached. So why is it important? Because before cell division and before the chromatids are distributed on the two daughter cells, we need to ensure that all the chromosomes have been aligned on the equator of the cell with no error at all. Now, what are checkpoints? What's the nature of the checkpoint? Actually, the checkpoints, all this mechanism is a very complex mechanism. It looks like an army of molecules. So how do molecules communicate with each other? What's the molecular language? The molecular language is through binding of molecules together. So all this regulation of the cell cycle that looks like having policemen, for example, that are standing by the cell and forcing it to stop unless otherwise mentioned. Actually, no, these policemen are nothing but proteins. So we have two families of proteins. We have the cyclins. These are the major proteins that control the cell cycle, and they are of different types, like cyclin D, like cyclin E, like cyclin B. But in the AP curriculum, we wouldn't bother you studying the differences between them. But why are we calling them cyclins? Because their level fluctuates throughout the cycle, and that's why we call them cyclins, because their levels are cyclic over the cycle, throughout the cycle. They increase and decrease in a regular fashion. And there is another group of proteins that shares the task with them, which are the kinases. And as you know, the kinases are also protein in nature, and the kinases are protein in nature because they are enzymes. And as you know, they are enzymes that cause phosphorylation by the direct addition of phosphate groups from ATP, or what we called before this in cellular respiration substrate level phosphorylation. This substrate level phosphorylation is not necessarily an activation. As you know, the substrate level phosphorylation for some enzymes, this is activating, but in other enzymes, it is an inhibitory, or it inhibits the action of the enzymes. Whether the kinases are activators or inhibitors, they are phosphorylators, and their action here, these particular kinases that regulate the cell cycle, their function is dependent on the cyclins. If the cyclins are not available, then the cyclin-dependent kinases cannot work, and therefore they are called cyclin-dependent kinases. These cyclins, as we said, increase and decrease. Their concentration starts to increase during G1 and then reaches its maximum, or keeps steadily increases during S, and then during G2 to reach its maximum before mitosis, just before mitosis. And then after mitosis, it starts to drop again. Cyclins, as we said before, have to bind with the kinases in order for them to be able to regulate the phases of the cell cycle. Assume, for example, that this is the protein molecule of cyclin, and this is the protein molecule of the kinase, the cyclin-dependent kinase. Then both together fit into each other, and when the cyclin reaches its maximum level just before mitosis, they enhance the cell to start the phases of mitosis, and that's why the complex of the cyclin-dependent kinases together with the cyclins is called the mitosis promoting factor, as we will be seeing later on. As we said before, when they bind together, the kinases become activated. They phosphorylate enzymes, and this phosphorylation might result in activation or inactivation of certain target proteins. These target proteins might be structural proteins, but they are mostly enzymes or functional proteins. But these mitosis promoting factors cannot work on their own. They have to receive signals. These signals are usually chemical signals, and they come from outside the cell and sometimes come from inside the cell. Because these signals should be received before entering into mitosis, so they are called pro-mitotic signals. And because they are coming from the outside of the cell, then they are called extracellular. And one of the most famous one is the growth factor. Growth factor, not the growth hormone. The growth hormone is a different entity. It is a hormone that is secreted by the anterior lobe of the pituitary gland. When the growth factor gives them the allowance to start working, they start doing what? They start preparing the cell for the S phase, because you might be wondering, haven't you said that the mitosis promoting factor promotes the cell to enter into mitosis, and mitosis comes after S and after G2. Why then are we saying here that the cyclin CD complexes promote the cell to enter the S phase? What you have to know is that the cyclin and cyclin-dependent kinases, they work together at different stages of the cell cycle. The most important one is the one that makes the cell join the M phase or enter into the mitosis phase. But earlier, this complex also enhances the cell to enter into the phase of the S phase, to prepare the cell for the S phase. Of course, if you want to prepare the cell for the S phase, then you have to promote the formation of the enzymes of DNA replication, and at the same time, because when you have a molecule of DNA and that molecule acts as a template for another molecule, you do this. Why? Because at the end of the day, you want these DNA molecules to be transcribed into, or part of them, every gene on the DNA molecule should be transcribed, meaning that we are going to use the sequence of triplet codes on DNA to form a complementary sequence of codons on messenger RNA. So you need the enzymes of transcription, and you need the transcription factors, as we said before, in molecular biology, to help the enzyme RNA polymerase to transcribe messenger RNA. So you're not only building up DNA, but you're also building up or preparing all the requirements for the next step, which is transcription of messenger RNA from DNA. But meanwhile, at the same time, they look forward to the future. They know that they have to be degraded at the end of the day. So they prepare the molecules that are required for ubiquitination. What is ubiquitination? Ubiquitination, meaning that if you have a protein and that protein is no more needed by the cell, then ubiquitin, or this particular protein, which is called the death protein, will bind to the protein, and ubiquitin will degrade that protein. So actually, ubiquitin does not degrade the protein directly, but once ubiquitin binds to that protein, that is, and I repeat, is no more needed by the cell, then the ubiquitinated protein is a labeled protein. So ubiquitination is a kind of labeling. We are like attaching a flag to that protein and telling the cell that this protein needs to be destroyed by a proteolytic enzyme. What is a proteolytic enzyme? A proteolytic, as you know, the lytic, or this suffix lysis, means digestion. So proteolytic enzymes are enzymes that hydrolyze proteins into amino acids. And now here we have a very important diagram for the growth factors. This diagram is present on the internet, and I found it as a Google image, but it is a very interesting diagram. And this diagram shows you that if you have a dividing cell, then the growth factor will bind to the molecule 1, and then 1 activates 2, and then 2 activates 3, and then CAM is activated, and RAS protein is finally activated to start the process of cell division. But meanwhile, the growth factor is capable of stimulating the cell to divide. So it is important for proliferation, which means rapid division of the cells. It is important, therefore, for cellular growth, and also in more complicated procedures for cellular differentiation, meaning that the cell would turn on certain genes, turn off other genes to have specialized functions, and this is differentiation. So the growth factor comes from the outside of the cell. So we can say that the growth factor is an extracellular molecule. But here we have another important, very important concept, which is contact inhibition, which means that if you have too many cells that are close to each other, then the natural response is to stop cell division. How does this occur? When there is contact inhibition, the CAM molecule, as you can see, extends across the cell membrane to the outside of the cell. And there is another CAM molecule that extends outside. Say, for example, this is cell number 1, or this is cell A, and this is cell B. Then the two CAM molecules will reach each other, will bind to each other. And when they bind to each other, there is like a disconnection between RAS and the rest of the molecules. And in such a case, when the growth factor stimulates 1 and 2, 3 is no more connected to M, and therefore RAS is not activated and cell division stops. So the mitosis promoting factor is a specific type of cyclin-dependent kinase complex, meaning that not all cyclin-dependent kinases are called mitosis promoting factor, and it is formed at the end of the G2 phase in order to promote the cell to enter the M phase, and it acts mainly, as we said, by phosphorylating multiple proteins. Why does it start working at the end of G2 and just before M? It's not only because its companion, which is cyclin, has reached its maximum before M, but because there is a phosphatase enzyme that acts at that time and removes an inhibitory phosphate group. See how phosphorylation is not always stimulation, because here when we remove the phosphate group by the phosphatase enzyme, the MPF got activated. This means that the phosphate group was causing inhibition to the MPF. This means also that phosphorylation is not always synonymous of activation. And the third piece of information that we have learned here is that the phosphatases remove phosphate, because what do phosphatase add? They add a molecule of water, so they hydrolyze the phosphate group and separate it from the original molecule. We talked before about the M checkpoint. But what happens at the end of every M phase? That MPF starts switching off. MPF starts self-regulation, self-down regulation. How does it? I forgot to tell you that although cyclin levels are fluctuating in the cell, yet the level of kinases are more or less constant, and we'll be seeing it in the next diagram. But why does MPF switch off? Because at the end of the M phase, we have to stop the cell cycle. Otherwise, the cell would continue dividing in an unlimited form without receiving regulation from the cell. Why does it switch off? Actually, it is because cyclin is degraded. So cyclin is degraded, the level of cyclin drops to almost zero, and therefore the level of MPF will drop to zero, although the other component, which is the CDK, is completely normal. And we will see this in this interesting diagram where we have two cell cycles, G1S, G2M, and then G1S, G2M. What happens is that the cyclin level starts rising during G1 and then throughout S and G2 to reach its maximum just before M, and when reaching M, it drops off. But the level of kinases is constant. The other fluctuation that you see, because when you add up something that is constant to something that is fluctuant, then necessarily the sum is fluctuant. So the red curve represents the mitosis promoting factor. The mitosis promoting factor rises during S and G2, not because of the rise in CDKs, because their level is constant throughout the cycle, but because of the rise in the cyclin level. And the MPF drops to zero at the end of the, not to zero, but to very low levels after mitosis, just because cyclin has become degraded. Maybe this diagram would show you more, because here we have G1 that gives, that makes, when G1 is complete, we enter into S, and then G2, and then into M, and this is the M checkpoint. As you can see here, that starting at G, the level of cyclin builds up until we are just before M. When the level of cyclin builds up to a very sufficient level, then at this time, it binds with the CDK to form MPF, and MPF will promote entry into mitosis. But afterward, they separate from each other. CDK remains in the circulation with its normal level, while cyclin is degraded because of ubiquitination. By having said this, we would have finished the video. Thank you so much, and see you in a new video.